So this is an inflammation of the heart valve. They travel through the blood, they attach to the heart. If you have an artificial heart valve, one of the things bacteria can do on these artificial valves is coat them with something called a biofilm. So we haven't talked about biofilms yet, but we will before the first exam. So very soon, we'll be talking about biofilms and how they form, okay? And so Staph aureus is the leading cause of endocarditis, and so the arrows here are trying to point to areas of inflammation in these heart valves. All right, so what did we talk about? So we said impetigo, scalded skin syndrome, abscesses, endocarditis. All right, and then I've got transmission, but before we're through here, I'll just tell you that Staph aureus also causes food poisoning. I bet you didn't know that. So because staph is on your skin, if you cook food, one of the things you do as you cook it is you tend to kill all the bacteria that are normally there. So you cook the food and then you handle it with your bare hands. And if you leave it out at room temperature, Staph aureus will start to grow because you inoculated it with your staph from your skin. And there are no other bacteria to compete for the space because you killed them, right, when you cooked. And then staph likes salt, so even if the food is seasoned and has some salt in it, the salt is staph like it. And when the staph grow, many of them can produce a toxin that causes food poisoning. And it's one of those things where a few hours after you eat, you're just miserably sick. So staphylococcal food poisoning is a really, really common kind of food poisoning. So I thought I'd throw one more illness out there that staph causes. All right, so transmission of staph. So 1 in 3 people carry staph in their nose. If that staph gets in contact with an infected wound, then that's an opportunity for that organism to get in there and cause an infection. So it tends to cause opportunistic infections. Another way you can transmit it is by sharing things like razors. If you share towels, if the towel has some staph on it from one person and you have a little cut, it's easy enough for the staph to get in there. So again, sharing equipment or supplies, particularly at a gym, is always a bad plan. Staph aureus, a lot of you saw staph last week under the microscope. And hopefully, if you showed it to me, I said, oh, that looks like staph. It might have been staph epidermidis because it looks the same under the microscope. And in fact, it's probably more likely to be staph epidermidis because pretty much everyone has that one. But staph aureus is maybe a third of people. But if you saw beautiful little clusters like this, they look like grapes under the microscope, and if you stain them with safranin or methylene blue, they would look blue or red. But if you stain them crystal violet, this is what they look like. Okay? So they're nice, round, very, very symmetrical bacteria. And you can even buy stuffed animals using staph. I actually have one at my desk. I have staph and salmonella at my desk if you want to see them. Oopsie. All right, epidemiology. So this one is, this is an important slide because what I really wanted to point out to you is that these are the 5 most frequently occurring bacteria in hospital patients, and staph is the first one on that list. So this causes a lot of skin infection in the patient. It's oftentimes staph. E. coli is another one, enterococci. Can't even see which one that is. Pseudomonas aeruginosa is this one. I'm not sure what that is. C-O-N-S. That's an acronym for something. So anyway, the take-home message here is that there is a lot of these Staph aureus bacteria in hospitals. And then when you look at the kind of bacteria that get into the blood and cause blood infections, bacteremia, Staph aureus is the second leading cause of those. So this is a big one. You won't get through a hospital career working in a hospital without seeing staph infections and blood infections as a result of staph. All right, how do we identify staph? Typically, it's a phenotypic identification because you can stain it so quickly and see that it's staph. It's pretty distinctive. If people have staph, we usually treat it with antibiotics like penicillin. We can use vancomycin if it's resistant to penicillin, but it gets tougher and tougher after that. If you have an abscess, the way they treat it is they actually open up the abscess and drain it. All right, and usually they'll actually put a little rubber tube in it, so they put a hole on 2 ends and put a rubber tube in there, so it will just drain until the abscess, the air gets in there and kind of dries things out, and the wound will heal. To prevent abscesses, you would obviously try to prevent puncture wounds. You know, sometimes you can prevent puncture wounds by, not using IV drugs, for example, but a lot of times when people get a puncture wound, it's an accident, right? So the main thing is to prevent behavior that leads to staph, but also, you know, bring your own towel to the gym, don't share razors, kind of things like that, right? A lot of people get staph infections, and the fact is if you're living out there in the world, you're going to get a few infections. Bacteria are everywhere. So I used to do these together, but it's kind of fun to talk about antibiotic-resistant staph in a separate lecture. So what we'll do is on Thursday, we'll talk about antibiotic-resistant staph. So today we kind of focused on the staph in your nose or garden variety staph because it causes enough problems by itself. Next time we'll talk about staph, what happens when it becomes resistant to commonly used antibiotics. That creates a whole new set of problems. Sound good? Yes? So this now we'll switch gears again and we'll talk about prokaryotic cell structure. All right, I'll give you a moment to regroup here as I kind of go back and forth. All right, one of the things I want to point out on this slide is this is a picture that we took not in our class, but a couple semesters ago. These are cheek cells, stained with methylene blue, the nuclei, and then these, we didn't see a lot of bacteria, at least in my morning section, but you can see the bacteria present on the cheek cells. So you're probably asking, why did I show this to you? Well, it's pretty enough reason. But actually, the reason I showed it to you is because we're going to talk about primarily prokaryotic cells today. But we also will talk a little bit about eukaryotic cells. And when I say eukaryotic cells are larger than prokaryotic cells, this is a nice visual representation of how much bigger a eukaryotic cell is than a little tiny prokaryotic cell. All right? Harder to sort of show this, I think, in a diagram. So you get a sense that you could pack a lot of prokaryotic cells into one eukaryotic cell. Eukaryotic cells are a lot larger. All right. So one of the things, once we sort of give up on the idea that we know how life came to be on planet Earth, because we know there's competing theories, and we're not sure which one is right, and there's evidence for all of them. At some point, we know that there was a last universal common ancestor, and that all life today has arisen from a cell that existed before that. So all life arises from pre-existing life. All organisms are composed of cells, and new cells arise only from pre-existing cells. What I want you to do now is get together in groups of 4 or so, and I'm going to give you an envelope that's got little strips in it of different structures, cells and types and structures. And I want you to work in a group of 4 and try to put them in the order of biggest to smallest. Yes? Okay. So I'll try to put 2 per row. See if you can get into groups of 4. If you have extra, you can get in between. Don't mix these up with another envelope. All right. If you need more, I really have plenty. So like I said, you're working on groups of no more than 4. And you have about 5 minutes, so you don't have to do a perfect job. Give it your best guess quickly. Try to get these arranged from biggest to smallest.
So a waste of food group. I guess. Never mind.
We have there 4.
Okay. Yeah, you I think a couple of the smallest.
Oh, yeah.
I think I'm interested in bacteria.
Yeah, the extra I think that's the right one. Cuban eggs.
green or
Mitochondria is another protein.
Yeah, that's right.
And amino acids.
sugar or protein.
What do you think?
Or you think
Another amino acids.
yeah, absolutely.
So this is the biggest.
I think that's the biggest because we can feel it in our eyes.
What?
Then travel solo me le it's big.
Oh yeah. Somewhere.
Después puede comer a los campos.
I think that this one right here is probably going to come before. And then the coli
My whole life.
Yeah, we're going to make them.
Yeah, the one, yeah.
But small.
Up, should go also.
How are you guys doing? You need a few more minutes? You ready to get the answers?
Exactly. And I think this is next to here. Bacterial, yeast, skin, and I don't know the exact name. I think it's pretty. Well, probably
How are you guys doing in the group? Okay, are you ready to see the answers? Yeah. Do you care more about them now? After you struggle? So when we start talking about the structures of prokaryotic cells and of eukaryotic cells, I think one of the challenges to try to figure out, what, because if we talk about little structures. You know, we just talked about monomers and polymers, right? In proteins and in carbohydrates. And so then you've got these big molecules, these polymers, and then the big molecules come together to form other structures, like for example mitochondria. And then those come with other structures to form cells. And so trying to get kind of a sense of everything in terms of dimensionality, I think can help your understanding of what the functions are of the different parts of what makes up living things. And this is a really cool interactive that's the University of Utah's genetics program put together. And so what I've got here are, and I've given you all the things in this interactive for you to kind of think about ahead of time and say, you know, what do you know, right? What do you already know and what don't you know? And then we'll go through it real quick because this is truly interactive. So here's a coffee bean. Here's regular 12-point font with a times font. There's a sesame seed, a grain of rice. And what this interactive allows you to do is drill down. So there's a grain of salt. How are you guys doing so far? Yeah, so, because you were doing these, right? You're trying to put them in order from biggest to smallest? Okay. And there's an amoeba. Remember we looked at amoebas and paramecium under the... They're pretty big, right?
Oh, what?
What?
Bigger than a human egg.
You saw them under the microscope, remember? Yeah? Okay, you don't, we're not grading you on this. This is all for fun. All right, and there's a paramecium. Does that look about right under the microscope when you saw them? Yeah, it does, doesn't it? Okay. All right, let's keep going, shall we?
Yeah.
All right, there's a human skin cell like your cheek cell. Okay. And here is a sperm and an egg.
Okay.
There's the sperm, there's the egg.
Are you going?
There's so much material here for late night comedy. The sperm and the egg. I think the main thing to think about with sperm and egg is sperm is a DNA delivery device. It deposits payload, it's done. Right? All sperm is basically a tail with mitochondria to power the DNA. The egg is actually really complicated because an egg has to, think of an egg is one cell out of the trillions that a female makes, but that egg actually has to jumpstart development. And so when an egg is produced and it becomes ready to be fertilized, it's not just half of the DNA. It's actually all of these proteins that are layered ingredients so that when the sperm fertilizes it, it triggers development. This one cell that's formed from fertilization has to develop into a trillion cells and go through the entire process of development and differentiation. And that process is triggered by the molecules that are ready to go in the egg. So that's why the egg is so much larger, because it actually has a much larger job in terms of development, at least initially. Okay? Kind of cool. All right. There's a red blood cell. Red blood cells don't have any nuclei, so they're definitely a little smaller. You'll look at your red cells under the microscope this semester too. Here's an X chromosome. If you like making bread, there's some baker's yeast.
Wow.
As we drill down further, how are you? I see people rearranging there. All right, that's good. That's why we're doing this. There's a mitochondria. This is an organelle. One of the things I want you to notice, if you have it, is how close in size a mitochondria is to an E. coli. So bacterial E. coli is actually very similar in size to just a mitochondria. Your cells might have hundreds of mitochondria in them, right? And I was showing you earlier, you can pass hundreds of prokaryotic cells with a eukaryotic cell. But there's actually a really important evolutionary relationship between bacteria and mitochondria. So I want you to notice that they're similar and then kind of tuck that away because we'll talk about it a lot next time. All right. Oh my gosh, there's even smaller things. All right, what's smaller than a bacteria? Well, viruses. Right? So there's measles, influenza, HIV, and actually there's another one in here too. There's hepatitis, which is a tiny virus. Viruses are not cells. Viruses are basically just DNA wrapped around with a protein coat, and then depending on the virus, an envelope on top of that. So we will talk, after the first exam, we will talk about viral structure. Viruses cause a lot of diseases. They're not very complicated, but they go into your cells and sort of take them hostage and take them over. All right, so viruses are the smallest things, and people debate whether or not they're living, but if you say they are, they're the smallest living things, and then we just have substructures. So there's a ribosome. Remember we said ribosomal RNA? is part of that structure. It's not all of it. There's a lot of protein there too. And then here's a transfer RNA. Here's a hemoglobin, just the protein. Remember the 4 subunits that form that quaternary structure? There's an antibody. That's another type of protein in the immune system. Here we go. There's a phospholipid, one tiny phospholipid that makes up cell membranes. And there's a molecule of glucose. There's a molecule of methionine, which is one of the 20 amino acids. Adenine is a part of the DNA or nucleic acid. And there is a single carbon atom. Did you all get that one right? All right, so that's kind of a nice interactive. We can go back. Ooh, bigger and bigger and bigger. I think that's such a neat interactive. Okay. So you can, in the notes, because you have access to these slides, if you wanted to show this to your friends and family, you could just click on this, right? And it opens up for you? All right, so just so you know you have access to that. All right. So it's 12:23. I have 2 more minutes, which I plan to use. All right. Yeah, you can put questions back in your books. So we're going to talk in this lecture set about the 2 different kinds of cells. So there are prokaryotic cells. Pro means before, karyotic means nucleus, before the nucleus, and then eukaryotic contains a nucleus. So these are going to, now you know that this is a terrible diagram, right? What's wrong with it? They're the same size, but then we know they're not. We know that you could fit hundreds of these into this guy. All right? So this is a prokaryotic cell, and this is a eukaryotic cell. Eukaryotic cells are typically much larger and more complicated than prokaryotic cells. When we go back to our 3 domains of life, thinking about eukarya and prokarya, those fall into the prokaryotic cell types. Excuse me, the archaea, I said that wrong, not eukarya. Archaea and prokarya are prokaryotic cells, and then the eukarya are all the eukaryotic cells. Even though archaea are more similar to eukaryotic cells in terms of the ribosomal RNA. Okay? And then let's just, we'll wrap up talking about just common bacterial shapes, and then when we come back, I'll show you some quicker questions on them. So we've already looked at cocci, those are the round bacteria, right? You saw these under the microscope most likely from your environmental samples. Maybe some of you saw rods, right? And then you probably did not see things like Vibrio or Spirillum or spirochetes. These are a lot less common, and sometimes you can't grow them in culture. When you have them in 2 at once, it's a diplo, so diplococci or diplobacilli. If it's in a strip, it's a strepto, so streptococci or streptobacilli. Some organisms like Micrococcus luteus like to form tetrads. So when you look under the microscope, you see these little groups of 4. And then if they form big grape-like clusters, we think of them as the Staphylococci. These are not the only kinds of shapes, though, because bacteria form all sorts of weird shapes. There are star-shaped bacteria. Here's some very square rectangular bacteria. Okay? So don't assume that those are the only kinds that you might ever see. All right. So I think the next question is a quicker question, which we will save for next time to see if you remember this, okay? Don't forget to give